Okay, so I'm now going to go into examples of microbiome research. Again, the goal being to illustrate how the tools work, showcase some recent data from around PennCHOP, and give you some ideas for methods you might want to use in your own research. So here's a study that we carried out with Gary Wu a few years ago. Fairly straightforward, but I want to show you how the analytical machinery operated in this case. So we're looking at the effects of diet and genetics on the murine gut microbiome. And this is an example of a 16S sequence tag study. So there are a bunch of mice, I think it's 5 in each group, a normal diet or high-fat diet, and there's wild type set and knockout set. And what they're knocked out for is realm beta, an intestinal signaling molecule that Gary's lab has studied. So we collected about 30,000 sequence reads, so pretty good coverage over the different samples. Each of these is a fecal sample where we extracted DNA, sequenced using 16S tags, aligned to databases, and read out the different bacterial phyla that were present. And that's what's shown in these stacked bar graphs. So in this one on the far left, for example, you can see it's mostly Bacteroidetes that's comprising this sample, a little bit of Firmicutes, a little bit of Proteobacteria. And we can dig down further in the phylogenies and assign sort of class, order, family, sometimes genus, sometimes species on the basis of these tag sequence results. So this is pretty interesting. You can see that it looks like there's a pretty big difference in the diets, with the blue more emphasized with the high-fat diet. That's Firmicutes and Proteobacteria. But by eye, it's sort of, you're sort of gazing at this and it's sort of indeterminate. Obviously, you need some way of collecting the output into a statistical framework and analyzing and asking about the extent of the differences. Furthermore, it's not that clear that there's anything going on with the knockout, at least as shown in these stacked bar graph representation. So a tool that we and lots of other people like to use is Rob Knight's program called Unif Rack. So I'm going to explain how that works and show how it operated over this data set, because this is a tool that we use pretty commonly and will likely come up in analysis over many samples and projects that people bring to us in the Microbiome Center. So the idea starts with the view that maybe if you're comparing two bacterial communities, you want to do so on a phylogenetic framework. That is, if you have two bacteria that differ between the two communities but are really closely related, maybe you want to give that a kind of modest score. And two bacteria that are from different phyla that are really different, maybe you want to give that a bigger score. So Rob's tool allows you to do that. So to compare two communities, what you do is align them on the same phylogenetic tree as shown here, and then ask how much of the branch length on the tree is shared and how much is unique between the two communities, and that gives you a score. So here you can see the red community is completely separate from this blue community, so all the branch length is unique, so your value is 1 in his Unif Rack measure. However, here you can see they're really mixed up, so much of the branch length is shared. So here the value is something like 0.5. They're much more similar. And so you can summarize the resemblance between two samples with a single number using this method. So you can do this with 3 sets of communities. Here's red community, yellow community, and blue community. You can see blue is a little better separated, and red and yellow are a little more mixed up. So you can do each pair and make a data matrix as shown here, where blue is more separate. So you can see the number is larger, blue versus red, or blue versus yellow, than red versus yellow. So you can make this kind of a distance matrix, and then there's a lot you can do with that matrix. You can do statistics over these distances, or you can boil out the data to make a principal coordinate analysis and visualize the resemblance of the samples, as shown here. So this is a space that's created from the data by mathematical tools. There's nothing special about this space, but the machinery is such that samples that are very similar to each other are plotted close together in this space, and ones that are more different are more distant. And now you can see that we see good separation on the first principal coordinate between the two diets, high fat and normal, but now we're seeing good separation also between the knockout and the wild type. And then we can do statistics over these distances and put p-values on the measurements and so on. So this shows you how you can take these vectors of counts of 16S tags and bring them to an analysis that allows you to do statistics, visualize distances, and explore for patterns in the data. Okay, so let me show you now a more complicated example, this being a shotgun metagenomic study over a large data set. And so this is the other extreme, a not at all simple study of human subjects. Here we are investigating the associations among outcome, microbiome composition, inflammation, diet, and antibiotics. This is led by Gary, me, Jim Lewis, Bob Baldesano, and a host of collaborators shown here. So we're able to investigate outcome, microbiome composition, inflammation, diet, and antibiotics. We studied 90 pediatric Crohn's disease patients who are initiating therapy, many of these CHOP patients, either of two treatments, elemental diet or anti-TNF therapy, longitudinal sampling of the gut microbiome, so now 4 time points over 8 weeks. So this is introducing a new twist here, which is time dependence also, along with the other factors we're trying to analyze. And so shotgun metagenomic sequencing, we analyzed over 500 billion bases of sequence information in this study. So four Illumina HiSeq runs, a really, really large study. So this summarizes some of the data at the first time point, just trying to get your feet wet here. So we take the reads from each of these samples. This is just the first time point, align to databases. And one thing you can do is read out the kinds of bacteria that are there. That's shown, each row here is a different kind of bacteria. Each column is a different sample. So here are the healthy samples, here are the control samples. Here's a whole bunch of metadata summarized up at the top. One thing we noticed early on, we meaning Eric Chen, who did all the analysis on this with Hongji Li, was you could cluster the samples into 2 different groups. So here's another kind of cluster analysis, akin to the Unif Rack thing I showed, though not identical, where we can see separation into this blue group and this red group. And that's shown up here. The red group is here, and you can see the composition of bacteria is rather different, often associated with things like gamma proteobacteria, which are often baddies, and quite different from the healthies here, which are dominated by Bacteroidetes. Another thing we could see in the shotgun data was there's a lot of human DNA associated with the more dysbiotic cluster, the sicker cluster, and that's shown here. Much higher proportion of human DNA if you're in this sort of sicker group. Also a strong association of these 2 clusters with antibiotic use, suggesting antibiotics were perturbing the community and perhaps allowing or associated with colonization with these adverse bugs. In this kind of data, though, we can also try to understand the genes that are there. And so this now is, each sample is a time 1 sample in the columns. Each row is a type of gene. And again, we see 2 clusters, one the more dysbiotic, as shown here in the Crohn's kids. The clustering is shown down here in the multidimensional scaling analysis. And we could actually sort the samples into the 2 groups quite effectively using information on what forms of genes were there. So we're able to interpret some of the gene pathways that are involved. Some seem to be important in antibiotic resistance. Perhaps nitrogen metabolism is another big player, as suggested by Gary. And so we have a bunch of sort of theories we can launch based on the sorts of genes we're seeing. And we're now trying to associate this with quite extensive metabolomic data that we've also collected. One of the more interesting things to come out of this data, and a real virtue of the shotgun approach, is it allows you to look at organisms that aren't just bacteria. So fungi turned out to be quite an important finding in this group of studies. So here again, this is time 1 data where here are the healthies, here are the Crohn's kids. You can see after a bunch of data filtering, we detected several fungal lineages with quite high representation. That's the more red and yellow in some of these samples. And so of these 5 lineages, particularly this dysbiotic cluster, the kids with the more messed up gut microbiome, we could see quite high levels of fungi. And that also correlated with the high human DNA. That's the red here. So we can see now a collection of features in these kids. Dysbiotic bacteria, high fungi, and high human DNA, also correlating with each other in the guts of these children. And so just putting it together, here's a summary, or one way of summarizing this vast data set. Here's the timeline. Here are some comparisons we could make at the different times. Disease early on, in the absence of anything about antibiotics, we could see some differences in bacteria, and they came in colonized with fungi early, even if you took account of antibiotic effects. Antibiotics themselves also promoted fungal colonization, and we could see some bacteria coming and going, including bacteria you want to not have around, increased associated with antibiotic use. The diet therapy had its own specific bugs that it increased, and it decreased the fungi. And inflammation we could assess independently at the end and see bugs that were increased or decreased associated with changes in inflammation. So a very complicated study yielding a whole bunch of new things to think about in terms of potential assays to track aspects of disease and possible therapeutic targets. So this is a really cool kind of study. I should warn you, in planning your own studies, we probably had statisticians working on this for a couple of years. This was a huge job to analyze all this data. And so that sort of brings up the point that the complexity of the study can have a big effect on what your costs are in planning something like this out, from pretty modest costs to hiring staff to carry out the analysis going forward. Another area of recent findings, recent activity in the field has been circadian biology meets the microbiome. We live on a revolving planet, and the microbiota has been recently shown by several groups, including Garrett Fitzgerald, to show oscillations on a 24-hour cycle. This is very nice work by Zhue Liang, working with Garrett. So here's two 24-hour cycles tracking mouse fecal pellets. Again, tag sequencing, aligned to database, bioinformatics. And you can see this the pattern of increases and decreases in Bacteroidetes and a reciprocal pattern in the Firmicutes. And they showed very nicely that messing up a gene that's important in circadian biology, BMAL1, caused a loss of this pattern. And another group has shown PER, another of the circadian genes, when messed up this kind of pattern also. So it's really cool. It may be associated with feeding. The Bacteroidetes are good at grazing on polysaccharides, such as those supplied by host mucus. So during lean times, they may do better by grazing on mucus than the Firmicutes, which flourish associated with a meal. And there's various other parts of microbiome research that would sort of fit with that kind of picture. But there may be more to it. It's probably not just food, according to the findings from this study. And Garrett and we are trying to extend this to humans now and have some pretty dramatic data in humans. Another area of research that is really promising and really exciting is the connection of the microbiome and drug metabolism. So the metabolic capacity contributed by the microbiota is vast, and there's more and more evidence that it's contributing to the metabolism of many drugs. So Peter Turnbaugh presented some very prominent data on microbiota and digoxin metabolism. Acetaminophen's also been shown to be influenced by microbiota. And again, Zhue and Garrett did a really nice job studying indomethacin, an NSAID. And we're able to show that if you take indomethacin, it alters the composition of the bugs that are in the gut. And similarly, having the microbiome or not alters gut metabolism. And they alters, indomethacin metabolism. And they were able to run this down and do a very nice mechanistic study. Turns out indomethacin is metabolized by gluconuridation, and that's a step in removal from the system. However, the gut microbiota can reverse that reaction. so that you recreate indomethacin, which can then be taken up again and recirculate. So actually you get a lot more drug exposure if you have the gut microbiota than if you don't. And so I think it's highly likely that metabolism of many drugs is going to be modulated by the microbiome, and that individuation of drug response among humans may well be a consequence of having different microbiota present. And so another area of microbiome research, and one that I think people are very interested in Penn and CHOP, is straight diagnosis using the shotgun metagenomic methods. So many of you will remember this report from the New York Times maybe 6 months ago, front page. The first use of metagenomic sequencing in diagnosis from UCSF. A kid with an inherited immunodeficiency had been very sick for a long time. Conventional assay methods had failed to reveal what the bug was. At UCSF, people took cerebrospinal fluid, sequenced, and were able to find an unexpected bug there, neuroleptospirosis. And upon treatment, the fellow got better, the patient got better. So this really sort of was the first example of model-independent shotgun metagenomics used to identify an otherwise occult infection and treat it successfully. So obviously there's a lot of interest in many locations in doing more of this, doing it systematically for patients in many settings. Here's one from my, one from my colleague Ron Coleman. This is diagnosis of intubated patients in the intensive care unit, a study led by Brendan Kelly. So in this case, each column is a sample. Each of these blocks indicated by dark lines is a patient, and so it's longitudinal from left to right for each of these patients. It's illegible, but these are different bugs on the rows. And in this smallish study, this is 16S tag sequencing, looking for bacteria that are present. In this smallish study, Brendan and Ron were able to find a number of infections in these patients that were not detected by normal diagnostic methods, including like an outgrowth of ureaplasma in a lady who died. Now, of course, these people were very sick for other reasons, but we were left with the feeling that we were seeing infections that the attending physicians would really want to know about. And so we're very interested in sort of developing this further and making it efficient with the long-term goal of trying to apply these methods to patient care. So how am I doing for time? A couple more snapshots, and then we'll get on to more of the technical stuff of working with the center. Here's a study from CHOP. Sick kids being studied for Crohn's disease, oral microbiome, study led by Judith Kelson and Kyle Bittinger. So there's a specific point I want to make here. Kyle and Judith were able to sequence from healthy kids, from kids with Crohn's disease, tooth scrapings, and find differences in the microbiota. Now, one kind of worries about just a one-shot study like that. You know, how reproducible would it be? Is this an important finding? So one step towards sort of challenging the importance is at least to investigate the reproducibility. And so they used a discovery cohort and a validation cohort and asked, when they did it all again with another group of patients, did they see the same kinds of bugs affected or not? And the answer was pretty heartening. They got quite good reproducibility over many of these. So you can see Campylobacter, Fusobacterium, Rothia, TM7. A whole host of characters were seen independently in two different cohorts. And so something to think about in, if you're trying to get going in a new disease state, trying to investigate some problem in your field, if it's possible, if you have the budget, to do a discovery and validation cohort, it's really a great way to do it. It's much more convincing if you do it all twice and get the same answer. And so we now have a number of bugs to think about as possible therapeutic targets, and also something that was really emphasized in these data too was the effect of antibiotics. We're seeing quite strong antibiotic effects as well. Human genetics and the microbiome. This is a really interesting area, a topic of a great deal of discussion in the field. There have been now several influential twin studies comparing monozygotic and dizygotic twin pairs, asking if the monozygotic have more similar microbiota than the dizygotic, which is a way of asking in healthy, functional humans, is there a strong genetic component? And initial small study said no, but then a much larger, more recent study showed a detectable difference between monozygotic and dizygotic, suggesting that even in normal, healthy individuals, you can see a modulation by the microbiome. In more extreme kinds of cases, it's easy to see effects of human genetic mutations, immunodeficiencies on the microbiome. There are multiple single gene traits that can affect the microbiota. And here's a partial list of some of the genes and some of the consequences of their mutation. And as you probably know, there are dramatic examples of breaking one or another part of the immune system with a mutation, leading to selective infection with very specific kinds of organisms. So you can have horrible disfiguring warts with HPV, human papillomavirus, if you're mutant in these genes, whereas if you're mutant in the interferon gamma or NF-kappa B pathways, you have much more severe infections with intracellular bacteria. And that's just a couple of examples of where breaking different parts of the human immune system by genetic mutation specifically influence different interactions with different members of the microbiota. And so really, any time you have particularly with immune genes, but with all human genes, I think it's reasonable to ask, what are the chances that a mutation in gene X is having phenotype Y by acting through the microbiota in doing so? Of course, not all of them will, but it's a sort of reasonable conjecture, a reasonable hypothesis at the start for many different mutations. So a couple slides on the global virome, which also can be queried using deep sequencing and is another wonderful topic for microbiome research. I told you about global bacteria. That was all sort of a straw man for telling you about global viruses. There's something like 10 to the 6th bacteria per mil in seawater. There's something like 10 to the 7th viruses per mil in seawater. Just enormous numbers. They apparently outnumber their hosts by something like 10-fold. And we think that that viruses outnumber bacteria in the human microbiome as well. So there's something like 10 to the 31 viral particles on Earth, making them numerically the most successful biological entities by far on the planet. So the human virome is similarly extensive and diverse. People are commonly familiar with persistent viruses that cause long-term infections, like the herpes viruses, papilloma, HIV, and HCV. We can be transiently infected with cold viruses, many other viruses. Our genomes themselves are some 8% viral, integrations of retroviruses that became endogenous in the primate lineage leading to humans. That's this line here. This is the UCSC genome browser. Here are repeated sequences, and LTR is these retroviral remnants. And then the bacteria and bacteriophage that prey on the bacteria of the gut microbiome are very abundant, very diverse. In fact, they're so diverse that the types aren't well represented in sequence databases. So if you sequence from fecal material, for example, align to databases, you'll commonly see a lot of reads that don't align to anything. And the inference is that many of those are bacteriophages that just haven't been sequenced and added to databases yet. So to study viruses selectively, you can do purification methods. For example, taking a stool, filter, put it through a density gradient that pull out particles of density expected for viruses, use nucleases to get rid of free DNA, then open up viral particles and sequence. And so it's possible to get virus-like particle preps and sequence those and analyze the viral cut from your sample. Many of these viruses are going to be bacterial, which are often not the first thing you want to study, but I think a very fascinating area. Bacterial viruses will define the composition of bacterial populations by predation, and transfer genes around by installing by integration and lysogenization, including many genes for toxins, antibiotic resistance, and other factors. So as I mentioned, these guys are hard to study because their genomes are mostly not in databases. They're kind of biological dark matter, and importance is largely uninvestigated, but a really fascinating problem, I think. So let me end this little bit on viruses with a study of animal cell viruses. This is, again, work with Ron Coleman, led by Jackie Young and Jason Christie. Here, the study is lung transplant recipients taking lung washes, bronchoalveolar lavage, purify viral particles, then purify DNA and sequence. And one of the things Jackie found was enormous levels of these anelloviruses, which are viruses that grow on animal cells, circular genomes, quite small. Here's a picture of them. These are almost ubiquitous. Probably most or all of you are colonized with anelloviruses, though you wouldn't even know they were there without the sequencing methods to detect them because they seem to be completely benign. They must be all through the blood supply. They're not known to be associated with any diseases, but boy, did we see a lot of them in the metagenomic sequencing. And then here's a validation using quantitative PCR, where each column now is a sample. Green is HIV plus, healthy HIV plus, well treated. Red is healthy people, and blue is lung transplant recipients. And so the y-axis here is a log scale. It's a little hard to see from quantitative PCR. And you can see many of the lung transplant recipients have much, much higher values. And so this is an indication that in this immune-suppressed state, these people are on lifelong immunosuppression. It's these viruses are inferred to have bloomed due to the immunosuppression, and you can see that in other settings as well. So this is just an indication of how you can take a disease state or a patient group and compare them to healthy, scan with shotgun metagenomic sequencing, and find a really interesting group of viruses growing out.